Hello, everyone. I'd like to welcome you to the Phytobiomes Alliance webinar series. We're very fortunate today to have Rishi Bhandari from Auburn University, he's a PhD student who is wrapping up his PhD this semester. But before we get to his presentation, I'll give you a brief overview of the Phytobiomes Alliance. So we are an international nonprofit consortium comprised of industry, academic, and governmental scientists. We have a number of Phytobiomes Alliance sponsors, which make it possible for us to actually do this webinar and to coordinate research at the international level. So just a reminder, phytobiomes is a definition for a complex systems of plant-based agriculture. It's the concept of a plant or phyto in a biome, whether that's soil or other substrates, and it's all of the interacting geophysical and biological components that interact with it, including management practices, because management practices influence every aspect of that. And some examples of phytobiomes can be everything from a vertical farm to a pasture to cropland as we know it, but also a forest and a vegetable garden, whether that's an urban garden or a rooftop garden. So our vision is that by 2050, all farmers will have the ability to use predictive and prescriptive analytics that are based on the geophysical and biological conditions for determining the best combination of crops, management practices, and inputs for a specific field or a specific biome in a given year. So just to give you a quick note about the dashboard, you can already click on the handouts section, so you'll be able to download already a PDF of my presentation, as well as a PDF of Rishi Bhandari's presentation. And please monitor the chat panel to see messages from the organizers, but we also often put links to papers that are mentioned in the presentation. You can be able to click right on that and see what paper is being referred to. And then also you can start submitting your questions almost from the very beginning. So there's a quick Q&A panel on there. I just also want to remind you that the webinar is being recorded and will be posted on the Phytobiomes Alliance YouTube channel. And usually we have that recording available by the end of the day. So it should be there by tomorrow for sure. And you can also sign up on the Phytobiomes Alliance on our website, as well as on our YouTube page, so that you never miss an announcement about our webinar series or any other workshops or events that we'll be having. And stay tuned also for the International Alliance for Phytobiomes Research conference next year, or International Phytobiomes Conference next year. It will be in Europe in the fall, and details for that are now being finalized. So we hope to get that announced very soon. So you can keep abreast of all that information by signing up to our mailing list. So at this point, I'd like again to welcome Rishi Bhandari, who is a PhD candidate at Auburn University, and he's going to be talking to us about phyllosphere microbial community structure. So Rishi, thank you very much for your willingness to give a webinar today, and we look forward to hearing your presentation.
Thank you. Thank you for the introduction. And I would like to thank the Phytobiomes Alliance for providing me this great opportunity to present my research. So this research is one of the chapter of my PhD, and it has been published. So today I'll be talking about how biotic and abiotic stress drive the phyllosphere microbial community structures, mostly focusing on pepper. So going briefly on my talk today, I'll be talking about the growth defense trade-off and how climate change induces this variability in the microbiome and the growth defense trade-off. And I'll be then talking about how the microbial community's response towards this climate change, and we'll be focusing more on the ozone as an environmental stress. So then I'll go on the knowledge gap and my experimental design, and then I'll be talking about my results, followed by summary and inference from this study. So as we all know that plants in their natural environments deal with the various biotic and abiotic stresses. And as the plants are sessile in nature, they do not have any other choice to escape from these environmental stresses. Rather, the plant has now developed a various mechanism in order to overcome these threats of various biotic and abiotic stress. So once they detect these stresses, they generate the necessary cellular responses. So as we see, like we can see various biotic and abiotic stresses here that the plant faces in their everyday life. But among the various biotic stresses, pathogen invasion is one of the most influential stress that affect the plant. The pathogen can modify the habitat by secretion of the various virulence factors, which will ultimately lead to the activation of the defense response in the plants. So the activation of the defense response results in the suspension of the growth, which is commonly known as the growth defense trade-off. And this trade-off has a profound implication for the agriculture and natural ecosystem, as growth and the defense are closely related to plant health and overall productivity. And here we can say that various growth modules that are present in the plant and various defense modules that are present in the plants. And once there is the activation of the defense module, there will be the lack in the growth modules, so which is generally referred to as the growth defense trade-off. Similarly, we have seen like the increasing evidences which suggest that microbial communities in the plants can balance this plant growth defense response against various pathogens. Although we saw that the pathogen might have a negative effect on the plants, but the interaction between the beneficial microbes that are present in the plants has been shown to influence the various benefit factors, such as the growth. It increases in the yield and nutrition quality. It helps in the tolerance against the various stress and resistance towards the disease and various growth promoting activities. So this role of the microbiome is well studied in terms of the plant rhizosphere and also in the human gut microbiome. But phyllosphere, which covers almost double the area of the rhizosphere area under study in the world, is neglected and understudied environment. In the past, we have seen like the various studies that have shown how the individual members in the phyllosphere microbiome and can contribute towards the plant health. But there are very few studies where the contribution of this overall microbial communities as a whole in the plant health and productivity is studied. And as we know that with the change in the global climate, it is predicted that the infectious disease can extend beyond its geographical and also the host range. So how would plant response and their associated microbes also respond to this changing climate and the emerging pathogen, because the pathogen is going to behave in unpredictable manners with the change in the climate, and balance the growth defense trade-off? A lot of this study has been done in regard to the rhizosphere microbiome. And as we know that rhizosphere microbiome is known as the first line of defense against various pathogens, climate change is known to be likely to alter the structure of these microbial reservoirs in the soil, which thereby create a microbial assembly in the will create a different microbial assembly and will allow the pathogen to cause more disease. Similarly, as we also know that plant maintains the homeostasis of the associated microbial communities via a tight and complex regulation of the immune system. The climate change can alter these immune systems response in which it may constrain the ability of the plants to assemble or to recruit the beneficial microbes, which in turns led to the it promotes the dysbiosis and finally led to the disease. Similarly, the cry for help strategy that has been well studied in the plants, which refers to the plant recruiting the beneficial microorganisms when the plant is under attack, either from the biotic or the biotic stress. And it is also likely to be altered under the climate change. So the climate change might constrain the ability of these plants to produce various signal molecules that can attract the beneficial microbes, or it might shift the microbial composition or their threats and their ability to respond to these signals. So with the recent advancement in using the genetic methods to determine the totality of the microorganisms and their interaction has shown how microbiome of the host influence the interaction between the host, the environment, and the pathogen. So we know that microbiome plays an important role in determining the overall plant health and productivity. So as we know from the previous slides, how climate change can affect the rhizosphere microbial communities and overall disease outcome, little has been known on how these changes may affect the phyllosphere communities and their interaction and overall plant health. So under the abiotic stress, as we know that the various ABA pathways are being induced. And when there is the biotic stress or the stress from the pathogen, there is an induction of the salicylic acid, jasmonic acid, or the ethylene pathways. And we know there is always a trade-off between these two pathways. So one of the area that has been understudied is when there is a combination of both the biotic and abiotic stress, how does it influence the overall plant health and how the microbiome, the phyllosphere microbiome, responds towards these changes. And as you know, one of the major threats, one of the major effort to control the plant disease is the use of the resistant cultivars. But with the change in the global climate, we know that the pathogen can behave in an unpredictable manner. And the current resistant cultivar might not be durable, as various defense pathways could be compromised during both the biotic and abiotic stresses. Among the various abiotic stressors, one of the most important abiotic stressors that plant experience due to the climate change is the elevated level of tropospheric ozone, which is the ground level ozone. The global warming that is caused by the greenhouse gases has resulted in the increase in the tropospheric ozone due to the rise in its precursors such as nitrogen oxide, carbon monoxide, methane, and other various organic compounds. In the recent year, the higher ozone level is being observed in the agricultural land around the globe, and it can be a potential abiotic factors influencing both the plant health and the microbiome. So as ozone concentration above 40 parts per billion is known to be highly phytotoxic, elevated ozone can negatively impact the plants in many levels. So one of the most common effects of elevated ozone includes the visible injury resulting in bronzing or reddening of the tissue, as we can see in this figure. It is also known to cause the reduction in the photosynthesis. Here, the two leaves on the left side are grown on the normal chambers or the ambient environment chambers. But when you look at these two leaves on the right, these are the soybean leaves, which are grown on the elevated ozone conditions. And this purple and the violet colors on these two leaves on the right, so there is a lower photosynthetic activities in these plants, which are grown under elevated ozones. And elevated ozone is also known to affect the plant growth, nutritional value, and crop health. And the influence of ozone on the crop yields differs based on the various crops, as we can see in here. So as we know that how climate change associated biotic and abiotic stress influence the rhizosphere microbiome and overall crop health, the important questions to address are how associated phyllosphere microbiome would respond to or contribute under this stress. So to address the question of the response of this phyllosphere microbiome towards biotic and abiotic stress, and especially focusing on host resistance, we use our model organism Xanthomonas perforans. So it is a foliar pathogen that can cause bacterial leaf spot in tomato and peppers, and it is an emerging pathogen of the pepper in the southeastern United States. So the figure in the right is based on the disease pyramid that I showed in my previous slide and is our overall experimental design. So here we are using elevated ozone as an environmental factors, which is altered due to the change in the climate. And we have two pepper cultivars which differ in their host resistance. One susceptible ECW, which is susceptible against this pathogen, and one resistant cultivar. This resistant cultivar is known to have um, the polygenic quantitative resistance against all 11 races of Xanthomonas ubasicatoria pathogen, which is the dominant pathogen of peppers in the Southeast United States. So as we know that quantitative resistance is known to be a stronger resistance as compared to the dominant resistance, and we want to study the effect of the pathogen. Here we have pathogen inoculated and controlled on this host resistance as well. So our overall study studies the effect of elevated ozone and the pathogen on host resistance and phyllosphere microbiome. So for our study, we started with the inoculation of the seedlings. The 5 to 6 week old seedlings from both the resistant and the susceptible cultivars were deep inoculated into the Xanthomonas perforans suspension. The Xanthomonas perforans suspension was adjusted to 10 to the power 6 CFU per ml. Similarly, if we look at the control plants were deep inoculated into the sterile MgSO4 buffer, and both the sterile buffer and the inoculum was amended with sylvate, which act as a surfactant. Once we perform the inoculation, we transfer those inoculated plants into the sterile 10-inch pot, and these sterile 10-inch pots were transferred into the chambers. So inside the chambers, we had 6 plants of each cultivars that were arranged one after another, and the control chambers will have the control plants, whereas the inoculated chambers will only have the inoculated plants. The whole experiment was conducted at atmospheric deposition site at Auburn University during 2021 growing season in the summer from May to July. Here, we harnessed the open top chambers that allowed us to test the effect of the ozone stress on the plant pathogen and microbiome interactions. These chambers, as we can see here, these are open from the top, so the plants inside receive the environmental condition is similar to the plants receive in the natural field condition. But we were able to manipulate the level of the ozone here in the plant's height. So we use total 12 chambers for fumigation, among which we had 6 of the chambers with the higher level of ozone, which is shown here with the red color, and we had the 6 other chambers with the ambient or the normal environmental ozone levels. Among the 6 elevated ozone chambers, the 3 chambers had inoculated plants, while the other 3 had the control plants. So it means like we have 3 chambers with 12 plant each. That means like 6 plants from each cultivars in each of those chambers, and they were inoculated here. The same thing here for the control plants, where we had 3 chambers with elevated ozones, where we had control plants from both of the cultivars. Similarly, under the ambient condition as well, we had 3 ambient conditions chambers where we had inoculated plants, 6 from each cultivars, and the same thing here for the control plants and the ambient conditions from 6 from each cultivar. So this is our overall experimental design. So looking at the ozone concentration, the average ozone concentration in the ambient environment was around 30 parts per billion, whereas the average concentration in the elevated chambers was around 90 parts per billion, and which is the estimated level of the ozone that is supposed to be reached by 2015 is per IPCC in the United States. So when we compare the ozone level between the chambers, the ozone level within the chambers in the control or the ambient chambers were not statistically significant, whereas there was a significant difference in between the elevated and the control chambers in terms of the elevated ozone level. So talking about our methods and shotgun metagenomics workflow, we sample the plants in the beginning, which we call as a base samples. Those were the samples that we took after inoculation or the control plants that were from the greenhouse. Then after the inoculation, we performed the sampling twice during the growing season, one in the mid-season, which is 60 days after transplanting, and another at the end of the season, which is around 90 days after transplanting. And the leaves from each of the cultivars within the chambers were pulled together to extract the metagenomic DNA, and that metagenomic DNA was sent for library preparation and sequencing under Illumina HiSeq platform. The raw reads were then trimmed for quality and contaminations, and the taxonomic classification was done using Kraken and Bracken. Similarly, the functional analysis of these samples were done using the human, and the network analysis to see the association between the microbial communities was done using NetComi. So this particular host pathosystem allows us to study the response of the resistant variety under combined stress. Thereby, we can assess the durability of these cultivars under the altered environment condition. But also, it also helps us to test the response of this emerging pepper pathogen, which is Xanthomonas perforans, on the susceptible and commercial resistant cultivars under this altered environment condition. So our overall hypothesis in this experiment that in the presence of the elevated ozone, there will be the increase in the overall susceptibility of both the cultivars against the Xanthomonas. Similarly, we also hypothesize that the phyllosphere microbial communities will show alteration in their both taxonomic and functional profiles with the change in the seasonal dynamics, and the response will be altered in based on the ozone labels. Another hypothesis we had was once there is the establishment of the disease, it would disrupt the seasonal dynamics in this overall microbiome, and this effect will be stronger in those environments where there is a higher disease as compared to that of the lower disease. So in order to test our first hypothesis that the presence of the elevated ozone will increase the overall susceptibility, we measure the disease severity index both during the mid-season and end of season. So if we look here, the disease severity index on the susceptible cultivars, we saw higher disease severity in the susceptible cultivars as compared to the resistant, which was expected because the susceptible cultivars were more susceptible towards the disease. But if we compare the disease severity between the two chambers that have the ambient environment and the elevated ozone, we didn't see a significant difference in the disease severity in these two cultivars, either in the mid-season or in the end season. But if we look at the disease severity on the resistant cultivars, we saw a significant increase in the disease severity both during the mid-season in the elevated ozone chambers and also in the end season as compared to that of the ambient environment. So this increase in the disease severity in the resistant cultivars in the presence of the elevated ozone shows that under this influence of the elevated ozone, there might be a erosion and the resistance in these cultivars. So as we saw the difference in the disease severity between the ambient and elevated ozone and the resistant cultivar, we wanted to see if this difference in the disease severity is the result of the change in the pathogen abundance and if the environmental factors has any influence on it. So we calculate both the relative and the absolute abundance using DNA concentration as a proxy on both of these cultivars for our pathogen Xanthomonas. And here we can see the red line which represents the relative abundance and the blue lines which represents the absolute abundance in the susceptible cultivars and then the resistant cultivars. And we saw the pathogen abundance increase from the mid-season to end season in both the chambers, but we didn't see a significant difference in the pathogen abundance between the ambient and the elevated ozone in both the susceptible and also the resistant cultivar. But one of the interesting thing we observed here was there was a high variation in both the relative and the absolute abundance of the pathogen population in the resistant cultivars in the presence of the elevated ozone. So to further confirm the influence of elevated ozone and the cultivars on Xanthomonas population, we analyzed in planta population of Xanthomonas perforans using a culture dependent method in day 7 and day 14. So while this short course experiment that we did within like 14 days might not reflect the outcome of the entire growing season that we did in our main experiment, but it allows us to evaluate the effect of this elevated ozone on the Xanthomonas population. So similar to the previous observation, we didn't see a significant difference in the Xanthomonas perforance population when we compare between these two chambers. However, again, in the elevated ozone and the resistant cultivars, we saw there is a high variability in the Xanthomonas perforans population, both during the mid-season and in season. So from this, the increase in the disease severity that we observe in the resistant cultivar under elevated ozone we saw that was not associated with the increase in the Xanthomonas populations, as we estimate either from the relative or the absolute abundance. So the high variability of the Xanthomonas population in the elevated ozone, both here in the relative and the absolute abundance and also in the population, might indicate a plastic response of the pathogen during adaptation to in these resistant cultivars when there is a change in the environmental condition, as we had higher elevated ozone here. So as we saw, the increase in the disease severity on the resistant cultivars and on the elevated ozone was not the result of the change in the Xanthomonas population. Then we hypothesized that this increase was associated with the significant reduction in the overall microbial density, which is associated with the resistant cultivars under elevated ozone, which suggests us that there is an altered prophylactic shield or the microbiota mediated protection from the microbiota mediated protection under this elevated ozone in these resistant cultivars. So comparing the microbiota density, we saw a significant effect of the inoculation on the susceptible cultivar, but we didn't see a significant effect on the microbiota density on the resistant cultivars, either in the elevated ozones or in the ambient condition. Next, we wanted to see the effect of the inoculation and the elevated ozone and their interaction on overall microbiota diversity and richness in these phyllosphere microbial communities. So overall, the bacterial richness and diversity values, both during the mid-season and end season in both the cultivars, were higher as compared to that of the base samples, which we can see from here. So this could be attributed to the low microbial colonization levels in the greenhouse grown base samples that increase in the diversity and richness upon exposure to the natural field condition. Then we look at the effect of the environment, the pathogen infection, and combined stress on microbiota diversity and richness. So in terms of the environment, we didn't see a significant effect of the elevated ozone on microbiota diversity and richness in both of those cultivars. However, the pathogen infection led to the significantly lower bacterial and eukaryotic diversity here on the susceptible cultivars. We didn't see a much significant difference of the richness in the richness in the resistant cultivars, but there was a difference in the diversity in terms of the resistant cultivars in terms of pathogen infection as well. Then, to visualize the difference between the bacterial and eukaryotic community structures between the samples and the two environmental conditions, a non-metric dimensional scaling was performed on Bay-Kurtosis dissimilarity by using cultivars, time of sampling, environment, and inoculation as an independent variables. And overall, we saw the effect of timing and inoculation was very significant in shaping the bacterial communities. Here we had mid-season and end-season samples were clustering together based on the samples as well. Here we have the susceptible cultivars, here we have the resistant cultivars, both in terms of the bacterial and the eukaryotic communities. And in terms of inoculation, there was a significant effect. Like here we can see there was a distinct clustering of the inoculated susceptible plants. However, the inoculated resistant plants behave as controlled susceptible plants. Then to look at the effect of the environment on the overall microbial community structures, an increase in the ozone level did not alter the bacterial community structure on the susceptible cultivars. However, it influenced the bacterial communities on the resistant cultivars in the absence of the pathogen. So here we found that the microbial communities on the resistant and susceptible cultivars were similar in the absence of any stress, but they were affected by the time of sampling, the inoculation, and the elevated ozone has an effect on the resistant cultivars in the absence of any pathogen. So to summarize my overall previous finding on influence of the environment, pathogen, and host resistant on microbial community structures, we saw under the ambient environment condition, there was no significant difference in the microbial communities, either in the susceptible microbial communities in the susceptible and the resistant cultivars. And there was a decrease in the microbial diversity and the richness both in the end season as compared to that of the mid season. However, in the presence of the pathogen was dominant on the susceptible cultivars, but it was not that dominant in the resistant cultivars, and there was a distinct microbial communities in these two cultivars. In the presence of elevated ozone, there was no changes in the microbial communities in the susceptible cultivars, and they were similar as we have seen under the ambient environment condition. But the microbial diversity, there was a distinct microbial communities in the resistant cultivars under this elevated ozone, suggesting that the elevated ozone influenced the microbial communities on the resistant cultivars in the absence of pathogen. And when there was a combined stress in the presence of both the pathogen and the elevated ozone, we saw the microbial communities similar to that as we have already seen in the presence of the pathogen. So we hypothesized that this change in the microbial communities, these microbial communities were similar as to the presence of pathogen, and so we hypothesized that under the presence of both the stress, I mean like both the combined biotic and the biotic stress, microbial communities might respond to prioritize their stress response. In here, the presence of pathogen might be a most influential stress, and they prefer to respond. The microbial communities prefer to respond towards the most influential stress. So as we saw, a distinct microbial communities across different environmental conditions, presence of pathogen, out of the combined stress, and we wanted to see how this distinct microbial community structures functions in the presence of this different stress, and how are they overcoming those stresses. So we wanted to see if this difference in the taxonomic difference also is being reflected in the microbial function. So the overall community function based on the relative abundance of the metabolic pathways as well as their associated gene families were mapped into these pathways were not affected by any of these factors. So this suggests that although there is the distinct microbial communities in the phyllosphere, they can perform the similar function in order to overcome this environmental stress. And this phenomenon of distinct microbial communities performing the similar functions has been referred to as a functional redundancy in the microbiome. So to summarize again, here from our taxonomic analysis, we saw distinct microbial communities across different biotic and abiotic stress. But in order to overcome these different stresses, we saw this microbial community this distinct microbial community performing the similar functions, which we refer to as a functional redundancy. So as we saw distinct communities performing the similar function, then we wanted to understand like how are these microbial communities interacting with each other in the phyllosphere, and how does this biotic and abiotic stress influence this microbial association. So as we know that plant cohabit various microorganisms, which includes diverse bacteria, archaea, fungi, oomycetes, protists, and various small insects, which collectively form its microbiome. And these microbes in the phyllosphere interact among themselves either in the positive or the negative way, and they form a complex network of interaction, which is known as the microbial network. And the strength and stability of this microbial network depends upon the various factors, and there are various parameters that are used to measure the strength and stability of this microbial network. And among those various parameters, modularity is one. So the modularity is the connection of the nodes within the particular module. So it means like higher the connectivity of the nodes within this particular modules, the higher will be the modularity of this particular association network. So the network that has higher modularity will have a dense connection within the modules, but it will have a sparse connection between the modules. And this is known as a more stable connection in terms of microbial interaction. Similarly, another factor we have is the average path length. So the average path length is the total number of the steps which has to be required to race from one node to the another. So if you look in here, the total distance from A to F, if we have a lower distance from A to F, it means like this particular average path length is less, and this association is more stronger as compared to that, like having a higher distance. Similarly, another parameter we use is the positive age percentage. The positive age percentage is the proportion of the positive age in our network. So here in this branch, if we look in the node A and E, if we look at the age in between this A and E, we see a green line, which means like a positive interaction. Similarly, if you look at the age here between A and F, we have a red line, which means like a negative interaction between these two taxa. So higher the positive age, it means like more stable the microbial community network is. So based on this various parameters, we look at our microbial network and how this different how does this various biotic and abiotic stress affects our microbial interactions and our microbial network. So if you look at the microbial association under the ambient environment conditions, we saw under this association we saw higher modularity, means like there is a more connection within the modules, and higher positive age percentage and lower average path lengths, which means like this under ambient environment condition microbial communities forms a very stable association among themselves. And if we see here, each of those dots represents the nodes or the particular taxa, and bigger the size of the nodes, it means like those particular taxa are referred to as a hop taxa on this particular network. And the hop taxa are those taxa which are connected to the most other nodes within this network, and they forms the core, or they form the stability of the particular network. So when we compare this particular network under the ambient environment condition to that of the elevated ozone, under elevated ozone, we saw a random network where we saw decrease in the modularity, decrease in the positive age percentage, means like there were more negative interactions, and increase in the average path length. And if we also see in here, if we look at the hop taxa that we see in here, we saw the different hop taxa under this particular abiotic stress. So from here we can see, like in the presence of the stress, the microbial, not only the microbial association is changed, but also there is a change in the hop taxa as well. So what happens when there is a presence of the pathogen? In the presence of pathogen, we saw more positive interaction and highly connected network with the higher modularity, higher positive age percentage, and lower average path length. And we can see like there is more positive interaction here in the presence of inoculation, in the presence of pathogen as compared to that of the control plants. And similar to the previous observation, if you look in here, there is distinct hop taxa as compared to that of the control plants in the inoculated plants. So it means that the plants are responding towards this particular stress by changing the interaction, but also by changing the hop taxa. So the higher positive interaction in this microbial communities in the inoculated plants might be just because when there is the presence of pathogen, there will be more resources available for the other phyllosphere residents. So they might be getting a free resources or the free food from the pathogen, and they might be having more positive interaction here. So as we saw, in the presence of the biotic stress, there is the distorted microbial community structures, and in the presence of inoculation or the pathogen, we saw a more connected network. So what happens when we have both of this together, like when we have both the biotic and abiotic stress? So in the presence of both the biotic and abiotic stress, we again saw the random network with the lower modularity, lower positive age percentage, and the higher average path length. And there is distinct hop taxa that are present here as compared to that of the control plants. So it means that in the presence of when there is both the combined stress of both the elevated ozone and inoculation, it again change the microbial association, and also the microbial network is random. So to summarize my overall talk, we saw like here, like talking about the taxonomic assignment or the microbial communities, we saw distinct microbial communities that are affected by the presence of pathogen, elevated ozone, or the combined stress in these two cultivars. However, these distinct microbial communities perform the similar function to overcome this stress, which we refer to as a functional redundancy. And in terms of the microbial association, we saw in the presence of various biotic and abiotic stress, there is change in the microbial interaction or the association. We saw distinct association between these microbial communities in these different treatments. And we also not only the association, we also saw the distinct hop taxa across these different treatments. And we assume that microbial communities under this different biotic and abiotic stress, in order to overcome those stress, they don't just change the interaction among themselves, but they try to overcome this stress by changing the hop taxas. So what are the two take-home messages or the inferences from my study, and what could be the future directions? So the most important finding that we found from our study is that host defense is likely to be compromised in the face of the climate change. And as we saw, the increase in the disease resistance in the resistant cultivar, which is known to be the most resistant, as it has a quantitative polygenic resistance. And there are various hypotheses that could be that can define our results. So one of them could be the altered host defense in the presence of the altered environment. So as there is change in the elevated ozones, the host defense might have been compromised, or there might be the altered defense, as I already showed, like how the ABA and the salicylic acid they have a trade-off between themselves. Similarly, another reason could be the pathogen being more aggressive when there is change in the environment. It means like the pathogen could produce various factors in the when it signals the change in the environment. And the other reason could be the loss of microbiota mediated protection, which I talk about the prophylactic cell. And in my study, I mainly focus on loss of microbiota mediated protections. So from our overall study, we saw the microbial communities in the presence of this different stresses, they just don't change their community structure, but also they change their interaction among themselves by changing the interaction, and also they change the hop taxa to overcome those particular stress. So in the future, as we saw the altered interaction among these community members, can this be used to offset the growth defense trade-off? It has been suggested that microbiome could be one of the main factors that can contribute towards the balance of this host defense trade-off. So can we restore these functions or the interaction of these microbial communities that we see in the change of this in the presence of this various biotic and abiotic stress, and balance this host defense trade-off? So as we saw various interactions across the biotic and abiotic stress, the synthetic microbial communities can be developed from the hop taxa that are identified across these various stress conditions, and they can be used as a microbiome mediated priming to balance this trade-off. So it's an open question that like what can be done in the future from our study. So with this, I would like to thank my major advisor, Dr. Neha Potnis, for giving me this excellent opportunity to work on this exciting project. My mentors for this project, Dr. Courtney Leisner and Alvaro Sanz, all my members from the current Potnis labs and past member Austin Holland, Auburn University for providing the funding and also the place to do our research, and NIFA for providing the funding for this project. This study has been published in ISHME Communications, as we can see here, and all our codes for this code and the raw data for this particular project has been deposited in this GitHub, which you can, if you want to access, the codes and the raw data are present in this GitHub. And if you have any questions or queries in the future, you can always reach to us on our social media account. And with this, I'll be happy to take if you have any questions.
Thank you very much, Rishi. A very interesting presentation. And we've already got a number of questions. But before we start the questions, I'd like to bring on Neha Potnis, who is the PhD advisor for Rishi, and who has been involved in this study from the very beginning. And Neha, do you want to make any comments before we go to the Q&A?
No, I guess looking forward to the questions.
Okay, great. And of course, the questions, I'll go back. If we don't get finished with all the questions, as Rolanda has already indicated in the chat box, we will be providing these questions on the YouTube page. And so you'll be able to see them in writing, written answers subsequently. All right, so for the first question, with regards to shotgun metagenomics, how did you mitigate host plant contamination?
That's a good question. Whenever we are doing the shotgun metagenomics, the host contamination is always a big issue. And so we have set up a set protocol for the metagenomic DNA extraction that we had published like on other publication as well. But the most important thing in taking the phyllosphere microbial communities and shotgun metagenomics is to have like less disturbance as possible. So if there is more disturbance on your host, it means like you're likely to get more contamination from the host. So what we do is like we don't try to disturb the plant, I mean the leaf, because most of our sources from our microbial communities as the leaf. So we don't, whenever we are trying to extract the DNA, we wash the leaf with the minimum disturbance to the leaves itself. And while also taking the leaf taking the samples, you have to be very careful, like don't break the leaf. I mean, like don't break the leaf too much. And whenever you're washing it, also minimize the minimize the disturbance, like the breakage of your leaf. And once we had the DNA, it is definitely not possible to get get rid of all the host reads, but we always align our reads, whatever we get, against the reads from our host. I mean, like in this case, we align the reads from our metagenomic reads against the paper reads using the need data. So you can do the host filtering using the program called the need data. So where you'll align your host rates against the against our metagenomic rates. And I would suggest to do like not just to use just one genomes or the reference genomes of the peppers, that while doing the host filtering, if you can do like a couple of different species of the, let's say for in our case peppers, to do the host filtering so that you will get rid of those host rates. In our case, we got almost like based on the samples, we just maybe like 10 to 15% at the most in our host reads, but some samples has had like 2 to 3% of the host rates.
All right, great, thank you very much. The next question is, setting aside all of the data from ozone treatment and the end of season data, did you look at mid-season data under ambient conditions to see if any specific taxa were enriched or present only in the resistant cultivar, certainly compared to the sensitive cultivar?
We look at like whenever we had both the mid and end season samples, we were looking at how the microbial community changes from mid season to end season, and is there the difference in terms of the microbial communities in the mid season as compared to the end season. But here we didn't show like, okay, what taxa were particularly increased in the mid season as compared to the end season. But we definitely did we look at the taxa that are difference in the mid season versus the end season. But in terms of the network analysis, we didn't do that. How does the interaction changes in between the mid season and end season because of the limited sample size?
All right, just to follow that up, do you have a recommendation on the number of sample size that might be available, that might be good to actually to get at that question? I mean, what would you do to increase your sample size?
in our case, we had a total of 51 samples. There were a shotgun metagenome. I mean, like we had 9 samples for each of those treatments at any particular point. So although we had 9 samples, it is still good to have a lot of samples. But we have seen like people doing the analysis based even like just based on the 3 samples or just based on the 3 replicates, right? But this type of analysis mostly focus on the strength of your the strength that you can say like, okay, whatever we have seen is the true in the nature, that was just true that's happening in the nature. So I believe that higher the sample size, it is better to do all this analysis. And although we have 9 samples, but it's still like some of the statistics fails even if we have 9 samples. So
I just want to add that it's very hard to answer that question at this time, as like what is the best sample size for this type of analysis, and especially with the network analysis, as we explored more, I think we learned more as to what are the limitations more so than of our inferences, when taking into account also even the eukaryotic community members. So the network that we looked at was only with the prokaryotic community members. But currently, I think we do not have methods that can address the eukaryotic community members and the cross-kingdom kind of network analysis with the shotgun metagenome data.
Yeah, that hopefully will be resolved in the future and become a possibility. I mean, I was very interested in the reality of the chaotic nature once you get into nature. You know, once you start looking at how it exists in real terms, it becomes increasingly random and increasingly chaotic, which then leads, you know, there are other tools that maybe we want to look at to make it more understandable. So, all right, for the next question, I just lost it. Based on your research, how do we prepare for climate change, and what should the focus of our research be?
yeah, that's a very interesting question. On our research, we saw just one abiotic stress, right? With what might happen when there is a change in the particular environmental factors, which we are supposed to get in the near future. So I guess in terms of the science in the futures, I guess more and more researches has to be done on like how does this abiotic change in the abiotic factors influence the pathogen, and also how does it influence the host resistance. And as we did this experiment, like whenever we are doing this experiment, we did this in one season, but in the nature it's being happening like the things that the change is being in the it's a slight change. So it doesn't change abruptly like from one year to another, like there is a just drastic change, and that's the temperature or any of those other environmental factors. So we'll see a gradual change, and I guess there will be also a gradual shift in this microbial communities towards this stress, and the microbial communities might also try to adapt towards this stress. But I guess more and more researches need to be done in course of times to understand how the microbial communities are changing and how are they adapting, and how the pathogen is changing, and how the host resistance are the how they are impacting the host resistance.
Yeah, so you're talking really about a lot of long-term research that will need to be done. But you also mentioned the synthetic communities. I mean, there is the possibility for the development of synthetic communities that could be used in the interim to try to address some of these situations. Would that not be true?
Yeah, definitely. So it's like in my last slide, I was showing like, okay, this could be the work in the future where under this different stress, we can always have those synthetic communities and try to overcome those synthetic communities. But in order to design those synthetic communities as well, we have to understand the behavior of those communities across these changes. So based on the long-term resources, researches, and whatever we see in the change in the microbial communities, and then we can definitely develop those synthetic communities and use those as a microbial priming methods to overcome those stresses. Is we know that the microbiome is the second genome, so definitely synthetic community is the one of the best approach to go with. But how to design the synthetic community is based on those researches, long-term researches.
Right. Speaking of those communities, how much within your microbial communities, roughly how much was bacterial versus fungi versus nematode, et cetera? Did any taxa respond differently to the treatments or that you saw?
So in terms of the total microbial abundance, definitely the approach that we use using the shotgun metagenomics, we had almost like 95 to 98% of the bacterial communities from our taxonomic classification. And so the approach that we use, Kraken and Bracken, we were not able to classify more of the eukaryotes communities. So we use a different approach. So that's why we were not able to make like the cross kingdom network there. But as we saw, like the dominating taxa, the phyla was the kingdom was the bacteria. There was very less in terms of the eukaryotes. So we didn't go well deep into the eukaryotes because their abundance was very less. I mean, like even less than 2%. And then whenever we want to extrapolate, like just based on the true percentage, that might be like very that might be not true as well. So dominating the way that we extract the metagenomic DNA is like we have the bacteria as a dominant, but we saw like 2 to 5% of the eukaryotes in the phyllosphere.
Maybe just to add, maybe yes, there is a bias, I guess, in our protocol as well, that we are sonicating the tissue and then getting the bacteria from that are attached to the leaf surface or inside. But maybe we are not doing a good job during that DNA extraction protocol of getting fungi represented well as they are in the natural samples. There were some taxa from bacteria as well as from fungi that were affected differentially in response to each of these treatments or each of these cultivars, but also interaction with the ozone environment. So there are some indicators that we have now if we are thinking of designing the synthetic community. Those that are most influential, as well as those that are hub taxa, could be the first ones that I guess we could go to.
Right, thank you. The next question takes us a little different direction. Davis and Aususpel, I hope I pronounced that correctly, showed in the 1990s that ozone induces salicylic acid biosynthesis and thus increased defenses against biotrophic pathogens. How did you consider these data in your system? Or did you? Did you look at this at all?
So maybe I'll go first with that response. So we do have a collaborator who is looking at gene expression studies on this particular experimental setup. That data is being analyzed at this time, but and I don't want to spill whatever their results are, but I think we have considered that. And if you look at the literature in the past, what has been done on effect of ozone on deciding the outcome of plant pathogen interactions, depending on the lifestyle of the pathogen or the strategy, the necrotrophic versus biotrophic versus hemibiotrophic, the outcome has been different. So the influence of ozone has led to either more resistance towards the pathogen or even increased susceptibility. And so I guess that depends on SA versus ABA pathways. And And again, I think we have to just wait until that data comes out, and then we will have more clearer picture as to whether we are really seeing ABA pathway being expressed more in response to the combined stress.
All right, thank you. The next question is, do capsaicin levels in cultivars have an impact on the microbial community structure and interaction? What's the current understanding regarding this, if there is any current understanding?
So talking about the effect of the cultivars on the microbial community structures, under the ambient condition or the normal condition, we didn't see a effect of the cultivars on the microbial communities. But in the presence of the pathogen or in the presence of the elevated ozone, there was a difference microbial communities in this two cultivars that we showed there. So it means like the cultivars definitely has a different response in terms of the microbial communities in the presence of this biotic and abiotic stresses. But to answer the question of the association or the interaction, we didn't, like based on our sample size, is we are biased with the limited number of samples, we didn't run a association analysis based on the cultivars. So we really don't know how the association analysis, how would be the association between those two cultivars, but we assume that based on our microbial community structures and as they were influenced by the biotic and abiotic stress, there would definitely be a difference in the microbial association between these cultivars.
And maybe I'll add to that is that our system that we used, or the experimental design that we used, we did not have near isogenic lines. We probably are not in a position to really address that question if the influence of resistance directly on the microbial community structure with this experimental design. We simply wanted to look at what would be the response of the commercially available disease-resistant pepper cultivar.
Great. Well, unfortunately, we are out of time. We do have a number of really great questions that have come in, and so these will be answered in writing and posted on the website. Both the I think it's on the Phytobiomes Alliance website, as well as at the YouTube, where our YouTube channel is. And just a comment about how we pick webinar series. It's mostly, I'll see an interesting paper and or someone will make a suggestion that they think that a particular study might be a great webinar. So if you have any of those, send them my way. I'm happy, or send them to Rolanda Young, or the organizer of the webinars, who takes care of all the technical aspects. And so with that, I'm going to sign off and thank again Neha and Rishi for a great presentation and response to the questions. And we look forward to seeing everyone at our next webinar. So thank you.
Thank you, everyone.
Thank you.